Good morning, friends. This is TransConnect, December 2023, and today we are going to talk about a very, what can I say, a topic which is debated so much. That is the Luminex donor-specific cross-match. It's got its good, it's got its bad, and it's got its ugly. So let us see where we can incorporate it in our algorithm, how we are going to use it in pre-transplant and post-transplant patients, where we can utilize it, and what are its lacunae and caveats, and where we should be careful and incorporating other tests so that it can give you the best results. So what is HLA? Let's start with it. HLA is human leukocyte antigen. It's a misnomer. Why? Because it's not just present in human, it's also present in other species. And it's not just present in leukocyte, it's present in other cells of the body. And it is interchangeably used as MHC. So some people call it MHC, some call it HLA, but for the sake of simplicity, we'll be calling it HLA. It's present on the chromosome 6 small arm, and it's got 4 megabase pairs, MHC region, and it contains around 140 genes, 20% of which have functions in immunity. So what are we doing? We are trying to detect the antibody-mediated rejections by doing this virtual cross-match or physical cross-match. One of the cross-matches, Luminex cross-match or Luminex DSA or Luminex lysate-based donor-specific antibody test, which we are going to discuss today. So there are three types of antibody-mediated rejection. We all know it can be a hyperacute rejection, which can be because of the preformed antibodies, or it can be an acute rejection, which can happen after days, and it can also be sometimes chronic rejection, which can happen after days and years together. So how do we detect this antibody? We have the HLA antibody screen or the PRA, which is going to give you uh, actually a thing that whether the antibody is present or not present. So if it is present, obviously, then there is some antibody. It could be donor specific or non-donor specific. PRA or panel reactive antibody is also a test which gives you a percentage of how much is the reactivity. So either of the two you can employ as a first test, and then you go for a cross match. Now cross matches can be physical cross match where you actually do the test like CDC cross match, Luminex cross match, and flow cross match. Today we'll be discussing about Luminex cross match, and you can do a virtual cross match by doing a single antigen bead assay on the patient's sera and also comparing it with the donor antigen profile or the donor HLA antigens. We can also test for non-HLA antibodies if your HLA antibodies are not implicated. So why do a cross-match test when screening seems sufficient? Antibodies against low incidence antigens are likely to be missed, acts as a mock transplant, and this was developed in an attempt to identify recipients who are likely to develop acute vascular rejection of a graft from a given donor. Now, it is mostly done, especially the CDC cross-match, for preventing the hyperacute rejection. In fact, it is a quality check for your laboratory. If your CDC cross-match is negative, more often than not, you should not have a hyperacute rejection because it is formed due to preformed antibodies which are in very, very high titers. And CDC cross-match can detect that. Similarly, the other cross-matches like Luminex cross-match or a virtual cross-match or a flow cross-match also detect donor-specific antibodies. But the point I'm trying to make here is these donor-specific antibodies may have a titer less than the CDC cross-match to be detected, but they are still in that much concentration that it can cause some kind of a rejection, either acute rejection or a chronic rejection. So the idea is to take off these antibodies, which are donor-specific antibodies. So what are the cross matches that are available? Different cross matches. First is CDC cross match, flow cross match. You have a lysate based Luminex cross match we are discussing. It can detect both class 1 and class 2 antibodies, and you also have a virtual cross match, which is a different topic altogether. So just talking about Luminex. Luminex technology, what is Luminex technology? It's a patented technology which was developed by Luminex. It is like an instrument, Luminex 100 or 200, which consists of series of different colored polystyrene beads. And these are synthetic polystyrene beads which are different colors. So like if you have mixture of two colors, they are in different proportions, and these colors will give you a specificity of each bead. So if like there is one bead, the concentration of two colors or the two dye is going to make that bead a specific bead, and each specific bead can then hold some antigen, and this antigen will be specific for that bead, so that when an antibody combines with this antigen, it is going to tell you that this bead is positive, which has this antigen coated on it. And when you measure it, and that how you measure it is mean fluorescence intensity. So when you measure the MFI values or the MFI or mean fluorescence intensity, you can tell that this bead specific to this antigen is positive or negative. So this is again a pictorial representation of the same. You can see there are different beads with different unique infrared dye concentration. So when you actually impregnate these beads with the dye mixture, you can actually demarcate between one bead from another bead. They're all 5.6 micron in size, and all of them have a unique specificity. So this is the instrument that I was talking about, Luminex. It's got this red and green laser. So whenever there is hydrodynamic focusing and the cell passes through this aperture, the laser hits it and that is captured by the bead detectors. So this is essentially like a flow cytometer, this Luminex instrument, and that is how it detects, and the readout is a mean fluorescent intensity. Now coming to the actual cross match, how do we do it? Luminex cross match. So basically you need, for any cross match, you need donor cells and patient serum. So for patient serum, you require a plain sample from the patient. You take out the sera. This sera, if it contains those recipient antibodies, they are going to combine with the antigen. And how do you get the antigen from the donor side? What you do is you take out lymphocyte. Lymphocytes are rich in both class 1 and class 2. So you make a donor lysate. So you add a detergent. So basically the cell wall is broken and the from the nucleus the HLA or the DNA material is going to come out, and then you have these capture beads. These capture beads are both present for class 1 and class 2. Now what this capture beads does is it's going to capture the HLA antigens specifically for class 1, and this capture HLA class 2 capture bead is going to combine with the HLA class 2 antigen. So basically it's going to capture the antigen. Now it acts like a material for an antibody to bind to. So once if there is a antibody present against this HLA class 1 antigen, it's going to combine with it and give you a positive result. Similarly, if the antibody is against HLA class 2, it's going to combine with this HLA class 2 capture bead, which has got an HLA antigen attached to it. So when there is an antigen antibody reaction, that can be read through a secondary antibody, which is AHG, like we have in the red cell serology, and this is read out in a Luminex instrument as a mean fluorescent intensity. Now the cutoff that we take is generally 1000 MFI for class 1 and class 2. So if it is less than 1000, the MFI values, it is considered negative. If it is 1000 to 1500, it is borderline or weakly positive. And if it is more than 1500, it is considered as positive. So it is a sensitive technique. Why it is sensitive? It's because of solid phase technology. You are using a Luminex based assay, and this has made the detection of HLA antibody very, very clear. But not many studies have been done on this Luminex lysate based crossmatch test. And why is that? Is because there is a there are some kind of a limitation that are present in this test which we need to know before we start employing it in our algorithm for pre-transplant testing. Right. So this is how a worksheet will look like. If you see, we have to run a control. So your controls should be all good. Your positive control, lysate control. I'm going to talk about this. It's got raw values, and then you have the adjusted value and final score, which is going to tell you whether it's positive or negative. So if you see, this is the lysate control that we employ with each test. You have these con values. These are backgrounds. This will adjust whether you have a lot of background or not, because each bead is going to give you some kind of a fluorescence. So you need to, take that into account. And then you have the tests. So 1, 2, 3, these three tests were the ones that were done. If you can see, the class 1 is negative here, class 2 is negative here. It is positive here because it is a value that is higher than 1000. And similarly, class 2 is also positive here. And in this test, both class 1 and class 2 are negative. You can see that. So depending upon your score, you can give your result whether it's positive or negative. So there are numerous quality checks that have been employed to make it properly functional. That is for Luminex Crossmatch assay. And that needs to be done because many a times we tend to evade these things, and that is going to cause a problem. And that is when your Luminex Crossmatch test becomes more vulnerable. So what do you do? I was talking about lysate control reagent, which I was talking about in the worksheet, if you see. So to know whether that HLA has been captured by the capture bead, you need to run an LCR, which is tested in parallel with the sera sample. So every time you run a test, you should have a LCR going on with it. So what is an LCR? It is a mixture of biotinylated antibodies that are specific for and bind to HLA class 1 and class 2 antigen. So basically what it does is tell you that whether the HLA is actually being captured or not captured. Now, it also, Luminex Crossmatch also has a control system to verify the entire assay is working properly. So we have a derived lymphocyte control, a negative control serum, and a positive control serum. All of this needs to be done at least once in a whenever you are performing a batch, or suppose you have a few batches coming over, so weekly or somewhere wherever you can incorporate it. If you think your assay is going fine, you can run a negative control or a positive control every time you change the kit. So that needs to be done, but only if your test is coming and correlating with the other tests that you are employing, like say flow cross match, single antigen, or a screen, that you need to do away with these kind of quality checks. But otherwise, mostly it is going to tell you whether your test is going fine or not. So, but all said and done, it's got its limitation. Most importantly, if you see, there have been many clinical studies that have questioned its role in the class 2 antibodies. So whenever you are trying to detect class 2 antibodies, you need to be careful. Sometimes it may give you a false positive result. So you need to know if your screen is coming negative. Suppose your HLA class 2 screen is negative and your flow is coming negative, but your Luminex cross match is coming positive. You need to be careful in class 2 specifically, because it may give you sometimes false positive results. The other thing is HLA class 1 Luminex cross match is more sensitive sometimes. So sometimes it may give you false positivity, and somebody may be denied a transplant just because your Luminex crossmatch is coming positive. So DR antibodies have been detected properly, but detection of DQ and DP antibodies have always been doubtful or absent. So in spite of this, HLA-C, DQ, DP, the test is considered very useful in a cost-constrained country like us. So we employ it as in part of our algorithm. So only after we did a few studies, and we have now got a considerable exposure in doing the Luminex crossmatch. Very interestingly, Bill and et al. Also tried to study the clinical relevance of Luminex crossmatch in a group of 165 renal transplant patients, and he found out that the sensitivity of Luminex crossmatch was in class 1 96%, which we also found out, but in class 2 it used to give around 35% false positive result. However, the specificity of Luminex crossmatch was 100% in class 1 and 97% in class 2. So this test has got its limitation, but then it's got the advantages as well. Why? Because it's a very inexpensive test. Viable cells are not required. We can measure the MFI value. We can have the baseline values. We can desensitize the patient. Again, do a Luminex cross match. The whole process takes just 3 to 4 hours. So in 3 to 4 hours, you are ready with a cross match. A clear distinction can be made between HLA class 1 and HLA class 2 antibodies. So you know which antibodies are implicated as a donor specific antibody. The donor lysate, especially in disease donor program, if you see, donor lysate can be preserved at minus 80 degrees Celsius for more than a year. So if you have this donor lysate with you, you can actually go for a post-transplant monitoring. So this is very important. Also, the ease of interpretation. Anybody can interpret the score that has been generated out of the worksheet. You can say whether this is class 1 or class 2. So very important that Luminex crossmatch is, if you have mastered it, you can employ it as a part of the algorithm or the test that you do for pre-transplant testing in, especially in renal transplant patients. So to conclude, Luminex Crossmatch can detect DSA with greater sensitivity than CDC Crossmatch. There is no doubt about it. Sometimes false positivity may occur. So you need to know your MFI value, especially for class 2. You need to have some supplementary tests. You should have, it's not a standalone test that you can, just do for organ allocation. And sometimes with other relevant tests, and when you combine all of these tests as a part of the algorithm, it gives a very useful information. So where is it in our algorithm? If you see, whenever CDC crossmatch is negative, we go for a screen, and along with that, we go for two cross matches, that is Luminex crossmatch oblique flow crossmatch. If one of them is positive, then we obviously go for a single antigen. If all of them are negative, go ahead with a transplant. So this is where the Luminex crossmatch or a flow crossmatch comes in your algorithm for pre-transplant compatibility testing. So this is our paper on the Luminex donor-specific cross-match, where we have evaluated the patients for both Luminex cross-match, antibody screening, and we also went ahead and did a single antigen bead assay, and we found that this is a very useful technique and a useful test, especially for the detection of anti-HLA antibodies in pre-transplant renal patients. More so for class 1, it can give you very good results in class 1. Class 2, sometimes it may give you a false positive results, but you need to be employing some more tests along with it. So thank you so much. I hope this small presentation on Luminex Crossmatch or lysate based Luminex Crossmatch was helpful. If there is anything you want to ask about this test or any other test that we need to cover, please feel free or write in the comment box. Thank you so much.